So we went through this again. This should be no question that we answered most of the questions, unless I said we're going to wait until to go over it again later, right? Someone asked, when are we going to do the pages that we skipped? We'll do the DNA replication. You're starting it this weekend, and we'll continue it after the test. And we're going to, right after we're done with DNA replication, we're getting to mitosis, meiosis, and then we're going to start doing genetics. We're going to start looking at Punnett squares, but complicated ones, dihybrid crosses, not single monohybrid crosses. And we're going to have to do FOIL. You know, FOIL, the stuff you're learning in Miss Simpson's class. We're going to have to do FOIL there to look up what our gametes are. And you're going to do all kinds of stuff. And so we're kind of behind. I'll be honest, we're a little behind. So I'm feeling a little stressed there. So we talked about plant DNA, bacterial DNA. We talked about the different kinds of chromosomes, how DNA is wound up, what the different structures. People were like, more than one kid came to me and said, well, how is DNA put away? How do you put away DNA? Well, this is a whole page here explained to you how to package chromatin. Chromatin is a word for the DNA molecule when it's unwrapped. So it looks like spaghetti when it's all unwrapped. The chromosome, as you know it, the little chromosomes we draw that look like this, the ones you cut out and taped on to the correct place on your page, they look like this, and they're different sizes and different shapes. This is actually what they look like here. This is a drawing. This is when they're all wrapped up and ready to be separated in the cell. This is for mitosis. They've been copied. They've been linked together, wrapped up, linked together, and then we're going to separate them. So chromatin is when the chromosomes are all unwound. We call them chromatin. When they're all wound up and packaged away, we call them chromosomes. But they're all the same DNA molecule. Are we all good with that? All right. So seeing as there's no questions and moving on, you look at the banding pattern, ask you questions, we answered them. Here's the activity we did in class. You did it, or you didn't do it, whatever. It should be ready to be turned in, right? This is not something you've turned in yet. There's what you cut. If you don't notice any abnormalities, you were supposed to identify which one was. All right. So if you don't notice any abnormalities in that was left blank, so that you can cut them out. Here's where you're supposed to pay. All right, just quickly, let me see, because someone asked about 2C. It says, if the arrangement is abnormal, state in what way and name the syndrome is displayed. All right, so in previous pages, there's a series of chromosomal abnormalities, correct? Have you read the rest of the other pages in the study guide? Yeah. So trisomy, we talked about trisomy 21. That means there were 23, trisomy 21 means you have 3 chromosome number 21s. What we call that is Down syndrome. So that's what causes Down syndrome, is you have 3 chromosomes. Instead of 2 here, there's 3 for chromosome 21. If you have 2 X's and a Y, you have another genetic disease. If you have 2 Y's and an X, you have another one. Oh my God, I want to say 2 X's and a Y is something called Klinefelter syndrome. The list is in there, right? Let me look and see, hold on. So if you don't notice any abnormalities, then you don't have to worry about it. But if you have 3, if you have 3 number 1s, then look it up. Say, what does it mean when you have 3 chromosome number 1s? If you only have 1 chromosome number 2, then what happens to someone who has only 1 chromosome number 2? You can look it up on Google, type it in, and it'll tell you the answer. In fact, do that right now. What is 3 chromosome number 21? Say 3, type it in Google right now. 3 chromosome number 21s.
Wait,
3 chromosomes. 21. 3 chromosome number 21s. What does it say? It should say Down syndrome. Down syndrome, I heard, right? Correct?
Oh yeah, it's right here.
All right. Now, what does it mean when you have 2 X chromosomes and 1 Y? Go ahead and look it up.
Say that one more time.
2 X chromosomes and 1 Y. I think it's Klinefelter syndrome, but we'll see. Type it into your phone.
Klinefelter
Klinefelter syndrome. There it is.
Is it sex syndrome?
Okay. So all you gotta do is if there's an odd number, it should be 2 in every slot, but if there's a weird number, chromosomes, look it up and see what and just Google it really quickly. It took you how long to do it? Literally, what, 20 seconds? 20 seconds? Yeah. So just write it in to them. So this is kind of the expectation we're at already. We did most of that in class. Can DNA really carry code? This page here, you have an entire case study in front of you. This is that case study. I like that case study. I'd like you to try to do it on your own over the weekend. It's interesting. If you want explanations, because you have questions, like someone just asked about pyrimidines and purines, you have 3 videos already posted on our class YouTube channel that we did in class. You can go over there, and we'll go over it, all right? But you trying to do it is going to make it so much better for you. So you have a 3-day weekend. You can get that done. And all the answers to that case study are already posted for you. 3 different versions of the answers posted for you on the YouTube channel. Is that clear? So there should be no reason why anyone finds this difficult. And obviously I go slow and there's all kinds of explanations. But if you read this, you'll see that these experiments that they discussed here briefly are discussed in detail in that study guide, in that case study. And then again, I explained it in the video. DNA molecule, what is DNA molecule? How long are they? What are introns? What are exons? We talked about this over and over again, cutting out introns, splicing together exons. This is all explained here. We went over it in class. I drew it out. I talked about post-transcriptional modifications. I love this because it gives you really nice detailed pictures. These are actual pictures from electron microscope of what DNA looks like when it's wound up like it is. Actually, this is actually chromatin, I believe. Somebody's gonna have to help me out. I can't. My eyes are old. Yeah, these are chromatin. See how it looks like spaghetti or rigatoni or something? It's all kind of unwound. So this is what I wanted to talk about today, because this is one of those pages that we said we're going to cover today, and I'd like to cover it. It's what are the different kinds of RNA? And what are the 3 that we've talked about so far you've been asked on in class? tRNA. What's rRNA? Ribosomal RNA, correct. What else? mRNA, which is carrying the message. And the transfer RNA, tRNA, somebody said over here and said, yeah, tRNA is another one. And that's the stuff that carries the amino acid. The transfer RNA is correct. So there are other kinds. It's one of those things like purines and pyrimidines. It's not something you have to memorize. The 3 that you have to know, we've already talked about. But you might ask yourself, how do we control genes? What do these other RNAs do? Well, this is a list of all the different kinds of RNA that are in your body, that you're in your cells, that do different things, and some of them are stuff that we do in laboratories. We use these different RNAs in laboratories to turn genes on and off, to cut genes up. So it turns out RNA has a lot of different functions, and obviously you have the mRNA, right? You have the tRNA and the rRNA. Those are the 3 we talked about, and the 3 that you really need to know. If you're not going to learn anything else on this page, you better know those 3. But then you have to modify the RNA. We talked about that, the post-transcriptional modification. Well, RNA gets involved in that. And it gets involved in regulating the translation, right? How do you regulate whether a protein gets made Or is it just the mRNA that's made, or is, how do you get rid of mRNA? How do you get rid of mRNA after you make it? What happens to it after you've made it, after you've made the protein? What do you do with the mRNA? Well, it turns out the RNA is used to destroy it. And then what do you do? And then we have these things called short interfering RNAs, or we also call them microRNAs. They interfere with the protein synthesis, right? That's how you turn off genes. You can turn off, let's say you have a protein, you're making a protein, insulin. I'm just using insulin as an example. I'm not sure if this is the control mechanism or not, but let's just say it's insulin. You ate a candy bar, so you made some insulin, got rid of all the sugar. Now you need to shut that off. You need insulin turned off. Well, one thing you can do is you could, again, not knowing how that particular mechanism works, you could shut it off. Obviously, you could stop making the mRNA, but you still already have mRNA that's made, correct? And that mRNA will continue to make insulin, even after you need it shut off. So how do you stop the mRNA from making protein? It turns out you have these things called microRNAs, that actually link up with the mRNA and stop it from being able to be used by the ribosome to make a protein. We can use this to our benefit. Why? Let me pause this. The point I'm trying to make is that there's two things you can do. You can slow down to a stop, that's turning off the gene, slow it down, and then get rid of the protein, get rid of the mRNAs over time. Or you can start to make these miRNAs, microRNAs that can stop the mRNAs from making a protein. So that's just one of many different functions RNAs do. So there's a bunch listed down here. There's 4 actually. There's obviously the messenger RNA, tRNA, and rRNA that you need to know for the test. I think I've said it 4 times now, so to make sure you know that these are needed for the test. Is that clear to everybody? mRNA, tRNA, rRNA. You need to know what they look like. What they do, not what they look like, but what they do and what their names are. Then you have RNA interference. So there are these miRNAs and siRNAs. They can be used to cut up RNA or to defense against viruses. When a virus starts to make its RNA in your cell, your cell can use these RNAs to shut down that virus. So these are things that your body can do. These are things your body can do to stop the production of proteins, for whatever reason. All right, that's all we need to see. Seeing there's no questions or concerns about the content, you need to go restroom, go sign out, take the pass. Then we can take a look at these ATGC, the base pairing rules. I think we should have this done already, right? This is pretty straightforward. I've seen a lot of kids already have it done. Please, again, in case you haven't done it, so you can have it ready to turn in next week, please remember that these are kind of shadowed out. You can just put them in. The T goes here, so obviously you're going to have an A here, or U if it's RNA. There were the ones you were supposed to cut out. This is left blank so that you don't put the back side up. Now, DNA replication, you're asking when we're going to do this. We're going to do this next week after you've done your case study. Read your case study. Read it because that comes before we actually go into detail of it. Then we're going to get into, of course, that's going to lead us to mitosis, meiosis, and then Punnett squares, gene inheritance, all the different ways of inheriting genes. This is the experiment in that case study. The video that you've been assigned that I've told you is called what? What's it called on the YouTube channel? Classic experiments. There's 3 of them on the YouTube channel. They go through these experiments. So I don't feel like I need to go over this in class. I think if you've gone over this experiment, You'll note, I want you to notice something when you're going over and doing this experiment. The reason we call it a classic experiment in biology is because it's elegant. It does everything an experiment's supposed to do. It sets up a hypothesis. It has control groups. It has beautiful control groups. It goes through and collects data and answers the question well. So make sure you're answering this stuff. These three you need to know for not for this test. Are you hearing me? Not for this test, but for the next test, you'll need to know conservative, semi-conservative, and dispersive. You'll need to know what those are. You'll need to know how does our DNA replicate. And so you'll do this activity class next week after the test. You'll be cutting it out. You'll be pasting it here, there. There's the three: semi-conservative, conservative, and dispersive. So if you do your reading, you do this case study this weekend, watch the video, you come in here on Monday or Tuesday, you take your test on Tuesday, by the way, right? So taking your test on Tuesday, and Wednesday, we're doing this, and you're feeling like very comfortable. You're feeling very comfortable. You're not feeling overwhelmed. You're feeling good about biology, feeling good about life. You can worry about any other class you want. You're ready to go. You don't do this work. You don't do this prep stuff. You can still do well. It's just you're not going to feel comfortable. That's all. It's up to you on how you want to manage your time. All right, we did this. Genetic code, correct? You answered questions. Now here's a question I'm going to answer here in the video. And that's the reason I'm going to answer it here is because people keep getting it wrong, so I want to make sure we get it right. This is 4. This is 4. And there's 4 here, right? So you get U, and U is phenylalanine. UUC is phenylalanine, right? So there's 4 possible combinations times 3. So 4 times 4 is 16 times 4 is 4 cubed, rather. 4 times 4 is 16 times 4 is 16 times 4? 64. There's 64 different combinations here. 64 possibilities. $64 possible combinations of the 3 letters, the 3 spaces, 4 for each, right? 4 here, 4 possible here, 4 possible here, leading us to 64 different combinations, possible combinations. Everybody okay with that? Do you understand what I just did? Because if you don't, now's the time to ask, because I haven't seen somebody get this right on their own yet. So you say you understand, but then why didn't you get it right? So here's the question they ask you. They say, if you did the reading, you'll notice that they said, look, we have 20 amino acids, and we got 64 different possible combinations. Well, that explains why we have doubles, right? And quadruples and 6 times, right? Because you have only 20 amino acids, but 64 possible combinations. That's why we call it a degenerate code. Okay, so I guess the question that they ask you here is how many amino acids could you code for if you only had 2 bases? So instead of 4, say ATGC, right? AUGC. What if you only had 2? What if you only had Z and Q? What if those were the only 2 bases you have? How many could you have? It would be 2 times 2 is 4, times 2 is 8. So there's only 8 possible combinations. Somebody say they're confused? Somebody say they're confused? Now's the time to say it. I can tell you, most of you didn't know this, because I've seen them. I graded this part, right? This part of the stuff you've already turned in. Of course, I haven't looked at all of yours, so maybe some of you got it right. So 2 bases, 2 possible combinations, 3 spots. So instead of 4 times 4 times 4, you have 2 times 2 times 2, which is 8. So there's 8 possible combinations. Then they ask you a follow-up question. Why is this number of bases inadequate, not adequate, for 20 amino acids. Another way of asking this question is, how come you need 4 bases? Why do you need 4 for the 20 amino acids? Why couldn't we just use 2? Why couldn't we use 3? Why 4? Right, so answer the question then. Answer the question I'm asking. I can't hear you. Because you can only, the max that you could do then is 8. If you only had 2 bases. I thought you guys said you weren't confused. So the maximum you could code for if you only had 2 bases in each spot is 8. And here you have 64 possible ones. So you could code for a maximum of 64, but you only need 20. So there's plenty. Here, if you're only doing 8, if you only have 8 possible combinations, 8 is the maximum number of amino acids you can have, but we need 20. So you can't have just 2 bases. That's why we have 4 bases. That's why we have A, C, G, and U. Are we clear? Those were the questions they were asking you. I hope that helped. Let's go through this thing called genes to proteins, the simplest case, right? The simplest idea of transcription. So here you have your DNA, here you have 2 DNA strands. One DNA strand has the coding strand, the other is the template strand. What does that mean, coding strand versus template strand? Think about it. Someone close that door. Go ahead, say it again. You're copying off a template. That's what a template does. You're going to use it to copy. That's right. Good thinking. So you're going to use it to copy. So you're going to use that. The template strand is what's used to make a copy of the DNA in the form of RNA. Are we clear? Only one of them is actually going to be used as a template, and that's this one. The one that says template strand. Which one? It depends on which chromosome, what region, etc. Now, this is a template strand. That means that the other side is going to match the code. This is the code. Where's that code? It's here. Notice, this is A. What do you think is going to be over here on this side? These two are bonded together, guys. Think. Use your mind. If this is A, yeah, this would be T, because that's what you're using this to match it up. This is the template strand, right? So where you see T here, you expect to see A here, because this is how this is made. This is mRNA. The question is, this DNA that the RNA polymerase used to read in order to make RNA had another matching strand to it, didn't it? It had 2 strands. It's double-stranded. DNA is double-stranded. So this code here should match this code here. Correct? Do you understand what I'm saying? Or are you just tired and just sitting there blanking out? Because you look like you're blanking out. Can you understand that if I have two DNA, a double helix, and here I have an A, T, G, C. Over here I have what? T, A, C, G. Whatever I have here, I have the opposite here. Not opposite, but matching pair. Correct? That we understand. I know you understand that. I know it. I've tested you on it. Okay, good. Now RNA polymerase is going to come down and read one of these two. Let's say that it's this one. Let's say this code here, this is the template strand. So it's going to read this. This is the template. What did you say? We're going to use it to copy. That's what somebody said. So that's correct. You're going to use it to copy. So where you see A here, what are you going to see over here in the template? In the actual mRNA? U. And here you can see T. What are you going to see over here? A, and then
C.
G. Do you see now that the template code matches this one, but this one is identical to this one?
Yeah.
This is identical to the code, except of course that instead of a T, you have a U, right? These two are the same. That's the same as what? The same as the code, right? So what are we going to call that other strand? The coding strand. We call it the coding strand because it literally matches the code on the mRNA. Okay? Does that make sense now? Why couldn't you guys just say you didn't understand rather than having me interpret your faces? It's fun. Okay. So here's a nucleotide. Remember that it has this nitrogenous base, it has that phosphorus backbone, transcription unit. So this is what's going to be transcribed, right? So here comes the RNA polymerase is going to read it. It's going to make a copy. All right. So this is your transcription. This is your mRNA from 3' to 5', correct? All right. In this is one mRNA, this is another mRNA, two different proteins. Here you're going to go where? What's going to happen here is this is going to go from where? Where's this mRNA going to go? To the ribosome. So it's this one. So you get two ribosomes. So you end up with two polypeptide chains, and you make this one thing called hemoglobin, right? One functional unit. Notice you have two proteins here. Two polypeptides making one unit. It's like having a tire. You have the rubber part of the tire and you have the hub, right? Correct?
Yes.
And then you have lug nuts. So you have a bunch of different things that come together to make a working tire. The same thing happens with any working protein. You can have many components that have to come together. This is one mRNA, this is another mRNA, two genes, one after the other. So everybody should know what those are. All right, moving on. So we should know, everybody had no problem filling in the start codon and the stop codon? What was the start codon?
AUG.
AUG. What's the stop codon? There's three of them, right? You can use the ones that are here. You can use the ones that we have right here. And what are the ones we have here? UAG and UGA. Those are both stop codons. Do you see that?
Yeah.
And the start codons are both AUG. They're right there. They're on the paper in front of you. So you should have those written down. What's the purpose of start and stop codon? To start and stop, to signal the start and stop of translation. Translation, not transcription. To end the polypeptide, correct. Start and end the polypeptide. The mRNA strand is described from which strand? Transcribed from the template strand, right? We get this? Anyone have any problems on this page?
No.
All right, not seeing any problems, seeing that we all understand, we're moving on. Do you need to know that for the test? No, you do not. Here's gene expression, prokaryotes versus eukaryotes. This is all based on what you see here. Describe the important features of gene expression in prokaryotes. What important features did you notice about prokaryotes gene expression? What's different about how genes are expressed in prokaryotes versus eukaryotes? Well, first, because prokaryotes lack a nucleus, right?
They together. They have to together.
Exactly. Transcription and translation occur together. So you don't have to they're not separated by a nuclear membrane, so they can happen in the same place. No problem. So that you can write that in here. That's the same as there's the answer there's 1. A. And RNA is translated into protein almost as fast as transcribed from DNA. The feature probably accounts for the lack of large amounts of intronic DNA in prokaryotic genomes. In prokaryotic gene expression, there is insufficient time to remove introns from protein coding sequences. So you don't have a lot of introns. That means there's no removing, changing things up. So that's the other one. So lack of large amounts of intronic DNA. That would be 2B or 1B, right? Everybody okay with that? So they're right there, 1 and 2. The traditional old view of gene expression, this is how we used to think that gene expression happened here. And then this is what we understand, how we think it happens now. So we used to think that all exons are made into RNA, and we used to think introns are junk. Introns are spliced out of the primary transcript. We used to think that, and we are pretty sure that that's still happening. Introns are being taken out, exons are being spliced together. But what we know today is those introns that we take out, they act as control mechanisms. They act as microRNAs, that stuff that sticks to the DNA and shuts down RNA polymerase or shuts down the protein, the mRNA. Yeah, those microRNAs, those introns can be used to help regulate the production of the protein. So that's one thing. And this model does not adequately explain gene expression in eukaryotes. So what model are they talking about? This idea that gene expression in eukaryotes was based on one gene, one protein. And why is it different today? Why do we think that that's probably not the case? The central dogma of biology is not correct? For a couple of reasons. How we post-transcriptionally modify the mRNA, how we cut up the mRNA is different. So you could have one gene leading to more than one enzyme. One gene could lead to more than one enzyme depending on how you cut up that mRNA. Where do you draw the line? Do you draw the line for the intron here or here? Make it longer or shorter? It all depends. Yeah. Yeah. So that's the key. That's why. The key is post-transcriptional modification. So in eukaryotes, the old view is still correct. In eukaryotes, because you don't have, the introns are not being cut out, if you have one gene, you have one protein, one enzyme. That's fine. That's how it works. But in eukaryotes, because we cut out and change the message, the code, after we make the mRNA, we can have all kinds of variations after it's been made into an mRNA. And I think we've already talked about how is gene expression in prokaryotes fundamentally different from gene expression in eukaryotes. We already talked about it, I think, at length now here in this video. So you should be able to answer that. Okay? Are we all good with that? All right. Does this making sense to you? Or don't just sit there and say, I know you've already answered these, but I want to make sure you get them. All right. All the rest of this, we should be... So next week, I think the test should be pretty easy if you understand these pages. This is just what should have been already submitted as your practice test, and we're going over these answers, making sure everybody has it right. Everybody should be able to explain what mRNA does. What is mRNA doing? Taking this message, what message? The message here from the teplin strand to the ribosomes out of the nucleus in eukaryotes, right? In prokaryotes, it's still taking the message to the ribosome. It just doesn't have to go anywhere. It's already there. Please remember, DNA is double-stranded. Notice that this is a eukaryote because it's linear. Remember, in prokaryotes, it's circular. Please notice that you have RNA nucleotides here for the RNA polymerase. Notice everything's here, all of it answered for you. Everything on the next page that's gonna, this you need, if you know this, that's like 90% of the test on Tuesday. If you know these steps, you're ready for the test. Explain the difference between the template and coding strands. I already did, right? The template strand is, right, this can be complementary to the coding strand. The coding strand is going to be identical to the mRNA except where the introns are taken out. They have the same sequence, except remember, what are we going to do after we make the mRNA in eukaryotes? We're going to cut pieces of this out. So we're going to change this sequence. That's what makes eukaryotes different. That's why that one gene, one enzyme concept is not as useful as it used to be. For the following triplets of DNA, determine the codon sequence of mRNA that would be synthesized. Here, triplet, this is going to make what mRNA? You should be able to do that quickly. Go. AUG. All right, so everybody gets that? I see a lot of people are bored.
Yes, a lot of people understand this. I don't know of anyone who doesn't. You've demonstrated on several different assessments, so I think we're good with this idea that when you have these triplet codes, you can read that codon chart and translate the take that DNA and turn it into mRNA. So that should not be an issue for anybody. How this DNA here gets turned into mRNA. And that there's this enzyme, this polymerase, RNA polymerase, that takes that code that's on the template strand and makes an mRNA. Remembering that the template strand is going to be complementary to the coding strand. In other words, that where you see a T here, you'll see an A here on the DNA strand, but as a RNA where you see an A here, you're going to see an A here. So the mRNA is going to be exactly that of the coding strand, except that where there's a T, there'll be a U. So that's just an orientation concept. So if you want to look at the code here, you can see that this code will be exactly this code here. You can take a look at the details yourself, where it'll be complementary to the template strand. All right, so you should be able to do this. This is a really good reference for you, as I've said in the past, as we said in class. So as you can see, the mRNA leaves the nuclear pore and goes out in the cytoplasm where it's going to get translated. Remembering that in prokaryotes, this all happens in the same place, so there's no need for it to leave the nucleus because there is no nucleus, right? All right.
Good. I will finish going over this with the other classes, and so you should get, you should have all the answers for the thing you turned in already done and ready for you to study this weekend if you need it. Also, I gave you some people understand it. Let's move on. Post-transcriptional modification. This is what we're talking about. This is the gene, right? There's three things you're going to do to this RNA. This is the mRNA. Initially, it looks just like the coding strand of the gene. It's identical to the DNA of the gene, except that, of course, where there was a T, there's now a U, right? Initially, it was exactly the same. But then the first thing that we do is we're going to start cutting out introns and sticking them together. We're going to decide how to stick these together. Now, if during splicing you take 1, 2, and 4 and you stick them together, that's one way of doing it. Now, this is the same gene. Is that correct? Do you understand that? The DNA is up here somewhere. I hope you're listening because I'm going to ask a question about this on the test. This is the same gene. The gene's up here somewhere. I made this mRNA. I'm going to cut this up into pieces called introns. Some things, some of the introns I'm gonna throw away. I'm gonna take the exons, I'm gonna stick them together. Now it's the same gene, but I can make 4 different proteins. I could stick 1, 2, and 4 together. 1, 2, and 4. If I cut everything else out, if I cut 3, and I splice everything out, I can splice and make 1, 2, 4. That's 1 protein. I can splice 2, 3, and 4 together. 2, 3, 4, or 1, 3, 4. That gives me another one. I could keep all of it, and then that gives me another protein. Or I could take 1 and keep it long, keep the whole of 1, or maybe I just take part of 1. The region 1. Region 1's here. Region 1 blue here. Region 2. I just cut out all of region 1 here, the white one. So depending on how I cut this up, I get 4 different proteins. Do you see that? That's how 1 gene could lead me to 4 proteins. Not 1 gene, 1 protein. This doesn't happen in prokaryotes often, because in prokaryotes, the transcription translation is happening at the same time, so the cutting out doesn't happen as much. 1 gene does equal 1 enzyme in prokaryotes. It's simpler. Here in this eukaryotes, we cut things up after we make the mRNA. So that also leads to a problem when somebody asks you, how many genes does a human have? Some people think it's 10,000 genes. Okay, it's 10,000 genes. Here's the problem. Even if you identify the genes on the DNA, that's right, we can have that same DNA sequence, we can process it differently and produce different proteins. So 10,000 genes could actually mean 100,000 genes. 1 million genes, who knows? 1 million different ways of processing the DNA. That's why humans are more complicated. Remember I said, do you remember our discussion of looking at the number of chromosomes? I said that corn can have as many as 108 chromosomes, right? And we looked at dogs have 78 chromosomes, I think, or something like that, right? And people were like, well, that doesn't make sense. A dog isn't as complicated, isn't as smart as a human. How come a dog has 78 chromosomes, humans only have 46? It doesn't make sense. Well, we do this really well. We cut things, we rearrange our introns and exons really well. We manipulate, we turn our genes on and off really well. We do a lot of this super DNA, above and beyond the DNA. We do a lot of this modification, so we can be very complicated, where prokaryotes really can't be. So it turns out that's one of our big differences between us and some of the things that are not as complicated as we are.
So when we look at the questions that you've been asked here, and again, it should be fairly clear as to what the answers are, but let's go through them. Explain why is it difficult to identify the true number of genes that are in the human genome. As we said in class, we know that there's 10,000 genes in the human genome, so humans, people think there's about 10,000 genes. Okay, well, that's fine. That means 10,000 what? That means 10,000 regions of the DNA that code for something, right? 10,000 regions that have that area that we just looked at through the whole, that have this region in front of it that controls whether the mRNA makes a transcript or not, also known as the promoter region. And then having this area here that codes for an mRNA, right? That has that whole coding strand and the template strand, and then you make the mRNA using an RNA polymerase, we just looked at in the previous slide and we talked about forever, and it's been the last 2-3 weeks on it. So this DNA molecule which we've also called a chromosome, right? Depending on whether it's wrapped up around histones or not, that DNA molecule has these little regions called genes. But because of what we're looking at here, because of this post-transcriptional modifications, because you can have post-transcriptional modifications, which we've talked about for a while now, right? This idea that after you make the mRNA, you're going to change it, right? You're going to take and you're going to cut certain pieces out. You're going to cut introns out, and you're going to take the exons and put them together. You can put them together in many different ways. So if you look here at how different ways of splicing, this one gene, this one area can lead to 1, 2, 3, 4, 5, 6. In this example, 6 different proteins, 6 different enzymes, if you will. 6 different translated regions, right? 6 different combinations. That's just in this example. So if you just did that, if all the genes could have 6 different possible combinations, you have 10,000 genes, you could have 60,000 genes instead of 10,000, 60,000 protein or transcripts, right? Or 60,000 different proteins from these 10,000 genes. So this idea of one gene, one enzyme is true in prokaryotes, but not true in eukaryotes because of this post-transcriptional modification. And it says, describe the ways in which mRNA can be modified to code different proteins. Well, we just talked about it, right? The introns can be arranged in different ways. You can skip exons so that you don't, so you end up putting different combinations, right? During splice exon may be skipped. So instead of it being cut up, right, it produces a different protein in mammals. Mutually exclusive exons, so in some cases only 1 or 2 exons, but never both will be incorporated into the mature mRNA. So again, look at the different combinations here. You can have, in some cases, you have 2, 3, 4, and 3, or you could have 1 and 3, 4, but you never have 1 and 2 together, right? So in any case, there's these 4 different ways in which you can have different proteins being coded for by this same gene. So depending on what's going on with any particular gene, you can have all kinds of different combinations. So the point is, it's difficult to know what the outcome is from a DNA transcript. It's not as easy as we would like it to be, as we hoped it be when they first started to decode this. Explain the advantage of being able to modify mRNA to produce different proteins. Now, again, what's the advantage? The advantage is that you can get a lot more function out of the same amount of DNA. Remembering the whole dog having 70 some odd genes or chromosomes, and the human only having 46, corn having 100 and some odd chromosomes, and humans having only 46. We get more complexity, more function out of less DNA or less chromosomes by having different ways of manipulating the DNA, having many more control mechanisms. We can have more differentiated tissues. So from the same DNA, right, we can have many different ways of producing many different kinds of tissues. So our brains can be more complicated. So our physiology can be more complicated. Right? So our function can be greater than the amount of chromosomes would make us think we should have. So this is very interesting that these ideas of post-transcriptional modifications can make that big a difference. So we're looking at translation now, and we've looked at it forever. And so you can see again, You have this tRNA. It's an RNA with an anticodon on it. That's the anticodon that matches the codon. That chart that you will have on the test lets you look at that. The tRNA is carrying. Notice it picks up an amino acid. This UUC will always pick up lysine. But there's different ways. There's different tRNAs that will pick up lysine, but this tRNA will only pick up lysine. So this anticodon will always code for lysine. The ribosome, which is made up of different subunits, the large and the small, they come together to make this ribosome. The tRNA matches, right? It brings in the amino acids. The amino acids then inside the ribosomes are put together in a long chain called a polypeptide chain. That amino acid chain is the protein. So when we look at codons for mRNA, you can see UAC, UAG, you should be able to tell me, and I know that you are able to, by the different assessments we've done in class and homework and classwork and projects. You've done it in class multiple times. You can take this codon, using the codon chart, find the anticodons, and from the codons you can also find the amino acid sequence. How many different tRNA types are there? Each with a unique anticodon. Well, you know there's 64, right? You know that because you've done the calculations for the homework. You know there's 64 anticodons, and there's only 20 amino acids. That's why there's more than one. That's why we call it a degenerate code, meaning that there's more than one way to code for the same amino acid. And do you know there's 64 different anticodons? Because there's 4 possible anticodons, right? There's 4 possible codons. That's U, A, G, and C, right? And there's 3 different positions, U, A, G, and C, U, A, G, and C. And that's that chart that we've looked at forever. And we know that if we take 4 possibilities times 4 possibilities times 4 possibilities, that's 4 times 4 is 16 times 4 is 64. So that's this number of various combinations of a triplet code that you can have if you have 4 different letters. So here you would have U, U, U. Here you'd have U, A, U. Here you have U, G, U. And here you have U, C, U, right? That's that whole chart that I'm not going to go into now, but you can go back and reference or look at your homework that you've done already on the work you've done already in the study guide, and you can see there's 64 different possible answers, 64 different possible combinations of codons. This is a codon chart, but for every combination of codons, of course, you have to have the complementary anticodons. So they have to match, right? So you have at least that many. Okay, let's go on.
If you know this summary, if you can fill this chart in, I'm not going to fill it here. I'm not going to fill it in here because it's really a good review for you. If you're watching this video and you just want to look at it, you can rewind it and look at the answers on the previous pages. You can look in the chapter. You can look them up. But guys, you should be able to see. A is DNA. Is the white one what? Which DNA is this one?
That's the coding.
That coding strand. Excellent. Notice the colors are similar. You see how this is light, this is light? This is the DNA strand that's called the template strand. But yeah, DNA, these are RNA, ribonucleotides, free nucleotides, however you want to call it. This is what? RNA polymerase. This one is what? The mRNA. This is the nuclear pores. This is the nuclear membrane. Yeah, you're finishing that by when? By Wednesday. If you have anything that you have not submitted to me this week, electronically or by hand, and you want me to grade it, except for yesterday's work I'll collect on Wednesday or Tuesday. If you have this work, these 2, I'm going to grade them this weekend. Make sure they're in this folder. Thank you for your cooperation. Have a good day. Yeah, I'm sick. Deal with it. That's why I was absent. Why'd you stay ill?
Because I was saying ill to you because I ill your presence.
You know you're recorded. I'll put that on YouTube.
Cool. Hey, everyone. Right. Y'all want to know my name?
Hey, Mama, you heard her say she's disgusted by me.
Watch your hey, Mommy.
And this is the last page you have to do for your review, as you know. So hopefully you have all these answers. So let's look at this. Using the word list provided below, identify each of the structures marked with a letter. Write the name of that structure in the spaces provided in the diagram. So the diagram on the opposite shows an overview of the process. And you saw the previous page as the diagram. Each of the structures involved labeled A through J, while your major steps in the process are defined with numbers 1 through 8. And you should be able to number all those things and name them. Here are the names of everything that you need to write in there, those structures, the nuclear membrane, the ribosome, the mRNA, the amino acids. If you need help with that, you can reference the previous classwork we've done. You can reference the page that has the answers on it, a couple pages before that. So you should be able to answer all these at this point. If you can do that, you're well prepared for the exam. Match each of the processes identified in the diagram numbers 1 through 8 to the correct summary of the process provided below. Write the process number next to the appropriate sentence. So you have tRNA molecules recharged with another amino acid. Well, what number, 1 through 8, would that occur? So you number them 1 through 8. You should be able to do that. You should be able to see that the recharging the tRNA means the tRNA is going to go get another amino acid. Where's that happening? What part of the process? What number part of the process does that happen? tRNA molecule brings the correct amino acid to the ribosome. Well, obviously this one's going to come before after the. Well, this is going to be recharged, so suggesting that it was charged. So this one has to come before that one, right? Unwinding of the DNA molecule. That's going to be inside the nucleus, right? So at what point does that happen? DNA rewinds to double helix structure. So for it to rewind, it must have unwound. So this one must come before this one. And you can see how just using logic, even if you didn't understand the process, and I'm sure that most of you do, if not all of you, and of course you have the answers here in the study guide and in the class notes. If you see that the DNA rewinds into double structure, then it has to unwind first. So this one has to come before this one. So even if you didn't do all that, you can logically deduce which ones would be first in numbers 1 through 8. DNA leaves the ribosome. It has to happen before you get recharged, right? So this one had to happen before this one, and this one had to happen before that one. Anticodon of the tRNA matches the correct codon of the mRNA and drops off the amino acid. mRNA moves through the nuclear pore in the nuclear membrane of the cytoplasm. That means that at this point, the mRNA was already made, the post-transcriptional modifications were complete, the DNA molecule was unwound, read, and then rewound already. So the unwinding, the rewinding already happened. And mRNA synthesis, nucleotides added, already happened in order for it to move out. So I think it's pretty simple to number these 1 through 8. I don't want to give you the answer here because you should be able to know what those answers are. But well, there's no but. That's it. Explain the purpose of protein synthesis. Obviously, it's so that you can produce the proteins that are necessary for function. Remembering that proteins are the functional units of the cell. They create structure. They run enzymes. They do everything that, or most things that the cell needs to happen. They're the builders of the cell. Name 3 different types of RNA involved in protein synthesis. Those are those 3. Now we know that there's more, right? We've talked about that, but the 3 that you need to remember are what? Again, you should know, but you should be able to easily say tRNA, mRNA at least, because especially since they're right here, tRNA and mRNA, right on the same page. Of course, there's rRNA, which is the ribosomal RNA. What are the 3 structural or functional differences between RNA and DNA? You should know what those are. Repeatedly, you've been asked to do that. How are nucleic acids attached to tRNA? Of course, what are we going to talk about that? We're going to talk about the tRNA and the RNA are going to match through base pairing. So the we're gonna have the anticodon and codon matching. Name the general process taking place in the nucleus. Transcription. Name the general process in the cytoplasm. Translation. Consult the mRNA amino acid table early in this workbook. Explain the result point mutation involving the change in the third base and nucleotide as follows. So if you have UUU and then you get UUC, what is that? What kind of mutation would that be? If you have UUU, what are the different kinds of mutations we talked about? We talked about point mutation, right? We talked about silent mutation. We've talked about frame shift. And frame shift happens when you have an insertion or a deletion. So think about these things as you move through and see if you can identify what these three are. All right. Well, good luck. And tomorrow's the exam, so good luck with that.